It's estimated that a typical pair of human eyes can see hundreds of different colors. But 23 million years ago, our primate ancestors were red-green colorblind. So how did we come to have the rich color vision we have today? I'm in Seattle to meet one of the world's experts on color vision, Jay Knights.
Oh, hey, Neil Shubin. Hi.
Great to meet you.
Good to see you. Well, great to be in the color lab.
Jay's been studying how animals see color for the last 25 years.
This is the place, huh?
Yep. This is where we test color vision in the monkeys.
Experiments conducted here show that most mammals, including many primates, are red-green colorblind.
This is Kramer.
Hi, Kramer.
And Kramer is colorblind.
Kramer is shown a red blob on a green background. If he kisses the red, he gets a treat.
If you can't see red or green, this is completely invisible.
Most humans can clearly see this red blob, but Kramer can't. He can't tell the difference between reds and greens. So why can't Kramer see the red blob?
Okay, Kramer, you are a star.
Kramer's eyes, like all eyes, rely on special proteins called opsins to detect color. They're held in thousands of special cells in the retina at the back of the eye. Kramer's got two types of opsin, each tuned to specific wavelengths of light. Signals from these opsins are then interpreted by the brain, which allows Kramer to perceive color. But to see color like we do, Kramer would need a third opsin, tuned to different wavelengths of light. We think our early primate ancestors were like Kramer. They had just two opsins as well. So how did they evolve a third opsin? The answer is in our DNA. Each opsin is encoded by a single gene. And when scientists compared these genes, they found that the gene for the newer opsin sits right next to one of the old ones. And significantly, they are incredibly similar. Both facts are telltale clues as to how the extra gene evolved. The old opsin gene was duplicated, and over many generations, one of these copies acquired small mutations that allowed it to detect different wavelengths of light. Could this change alone have really driven our ancestors' color vision? To find out, Jay set up another experiment. He implanted a third opsin gene from a human directly into the eyes of a colorblind squirrel monkey named Sam.
What we did is really a test to see what's the minimal thing you could do in order to give an animal color vision.
The results were astounding. Sam used to fail this test. Now he can easily tell the difference between reds and greens. Jay has recreated evolutionary history and given Sam human-like color vision.
You might think, oh, it would take them a long time to learn this new pattern in their brain. But as soon as the gene was turned on, the animal began to make these discriminations that they couldn't make before. And so in one, very short evolutionary step, it goes to this totally different world. You go from just having strictly, let's say 5 colors, gray, black, white, blue, and yellow, to hundreds of different colors that are all the blues and greens and purples and oranges.
One simple shift opens a whole universe of...
Yeah, that's the amazing thing. It's a multiplicative effect.
The ability to see a broad range of colors made it easier for our ancestors to find ripe fruits and young leaves. They passed their color vision to their descendants and eventually to us. We owe our rich color vision to monkeys living in an ancient forest. Walking on two legs marked a huge change for our species. In fact, being almost exclusively bipedal makes us unique among our primate cousins. The question is, how did our ancestors start spending so much time on two legs? A fossil found in Ethiopia revolutionized our understanding of this transition. I'm in Berkeley, California to meet Tim White, one of the leaders of the team that discovered Ardipithecus ramidus, Ardi. The team was digging in the Afar region in Ethiopia. Then, in 1994, a graduate student named Johannes Haile Selassie found a fragment of hand bone in 4.4 million-year-old rock.
He picked up and said, "This looks like a hominid." So we scraped and brushed that surface, and we found some of these hominid hand and foot bones in place. And so we focused on that little hill.
This was a hugely significant find, a new species of hominid from a time when scientists think our ancestors were becoming bipedal. It took experts about 15 years to fully piece her together. Biologist Owen Lovejoy has been figuring out how Ardi might have moved.
The critical thing in bipedality is the structure of the pelvis.
The pelvis was crushed during fossilization. From its reconstructed form, the team was able to deduce how Ardi likely moved.
This is a plastic structure that's produced by a three-dimensional printer.
Cool.
The top part of Ardi's pelvis looks more human. The hip bone is short and broad, a key indicator of bipedalism. But the lower half is much longer than a human pelvis, more useful for climbing. It suggests that Ardi was a creature in transition. She could walk on two legs, but not as well as you and I. Her feet support this idea.
This thing had full grasping ability.
So she has a grasping foot that can walk.
Yes.
It was a mosaic of anatomy that had never been encountered before.
So while not as nimble on 2 feet as we are, Ardi was a much better climber. But it wasn't just her body that came as a surprise. It was also her environment. The team collected around 150,000 additional fossils. Wherever they found Ardipithecus bones, they found woodland creatures, parrots, monkeys, and peacocks, as well as woodland plants.
It was a woodland, not an open savanna setting.
Now that's a
These
surprise.
are woodland animals.
That runs counter to so much of
It
what
runs counter
people
all the way
expect.
back to the 1800s.
So now we have a new, detailed picture of how our ancestors made the transition to bipedalism. Ardi was a woodland creature. Small, just under 4 foot tall. She was a good climber, using all fours when in the trees. But she walked upright when on the ground. Our ancestors started walking on 2 legs while still living in the woods. Ardi suggests that by 4.4 million years ago, the transition to walking on 2 legs was already well on its way. Our ears allow us to pick up a wide range of sounds. From high-pitched squeaks to low, long-distance calls.
Okay.
The secret to their capability lies in the tiniest bones in our body.
These are the 3 bones of our middle ear. All mammals, including people, have these 3 bones that sit inside the skull. And what they do is they form a little lever system that turns the vibration of air into sound that our nervous system can perceive. We, like all mammals, have 3 of these bones in our middle ear, but reptiles only have 1.
These 2 extra bones, the malleus and incus, enable us to amplify sounds, which is why our hearing is so much more sensitive than most reptiles. This presents us with a simple yet extraordinary puzzle. How did our acute sense of hearing, using 3 ear bones, emerge from our reptilian ancestors who used only 1? For nearly 2 centuries, scientists like Karen Sears have been fascinated by this mystery. She's one of my former students studying evolution by observing the development of these possums.
So what we see in the opossums is from that time they're born through their adults, they almost go through 300 million years of evolution in terms of their ears.
By looking at each stage of development, Karen has evidence of how our ear bones evolved.
So what's really surprising is if you look at these early opossum embryos, what you see is essentially a reptilian style ear with 1 ear bone. Those 2 extra middle ear bones, they're going to be in the ear in the adult. They are part of the jaw. And so the jaw joint between the skull and the jaw really looks like that of a reptile. And then after they're born and while they're growing up, those little bones that are going to be the mammalian middle ear disconnect from the jaw as they get relatively smaller and move up to their final position to become a mammal-like ear. So if you have modern reptiles with 1 ear bone, you have modern mammals with 3 ear bones. I think what the embryos do is they almost provide a link between those 2.
So how do we know that the bones we use to hear with came from the bones reptiles used to eat with?
Well, this amazing transition we see in development from jaw bones to ear bones, we also see in the fossil record. And we see it fossils from here in South Africa. And if we look at some of these primitive mammal-like reptiles, we see something in the lower jaw that's very different from us. Because what it has is 1 bone here which carries the teeth. And then there's a bunch of other bones that are sort of moderately sized that sit at the back of the jaw. They form the jaw joint itself. As you go forward in time, what you start to see are creatures like this, which have a larger bone that holds all the teeth, and ever smaller bones at the back of the jaw that form the jaw joint.
When we get to animals that lived around 200 million years ago, more recent in the fossil record, what you find is that they've formed a completely new jaw joint. That's because the bone that holds the teeth had grown so large it made contact with the skull. So over millions of years, bones that formed the old jaw joint began to shrink and eventually became redundant. But instead of being lost altogether, these old bones were repurposed to take up a new role in the ear. The story of our hands can be traced back to when ancient creatures like Tiktaalik used their fins to crawl out of the sea some 375 million years ago. Ancient mammals modified those fins into paws. Creatures like Hadrocaudum had short fingers that all pointed in the same direction, tipped with claws. Then, a hand appeared that was very different, and we can see it in a tree-dwelling species called Gnatharctus. This ancient hand can tell us a lot about our hands today. I'm meeting fossil hunter Jonathan Block. He has a pretty complete skeleton of Gnatharctus, a primate that was first discovered in Wyoming Territory in 1870.
Wow.
These are the real
this is the real deal. That is absolutely exquisite.
This is a 50 million year old primate skeleton. It gives us a window straight into the world of what the earliest primates would have been doing, how they would have been interacting with their environment.
Like most modern monkeys, Gnatharctus was a climber, adapted to life in the trees. The evolution of this creature, and others like it, had a huge impact on one of the features that most defines us: our hands.
The thumb is divergent, that is, it forms an angle with the index finger. And so that shows you that it could bring its thumb into opposition with the rest of the digits.
Nitharctus also has long fingers and nails instead of claws. This is one of the first times in the fossil record that we see a hand that looks like our own, the hand of a primate. It's a turning point in the history of the human body, an anatomical change that would eventually let us shape the world around us. But why did primates evolve such a hand in the first place?
Okay.
To find out, I'm going to look for clues where my ancient ancestors lived, up in the trees. Thankfully, I've got ropes.
Okay, ready? Yeah. Got it, natural. Woohoo! A little naughty up here. Okay. Woohoo!
These creatures are so unbelievably agile all through here. This is their home. They are able to live on small branches, big branches, throughout the tree. They can jump branch to branch. You know, when you're in the tree, what you see is that the canopy is not one place. There are lots of environments, lots of niches up here. And one special one is on the ends of the branches, because it's there where the flowers and the fruits and the insects are. So the rewards are great to be out there.
We call it the fine branch niche, and for the earliest primates, being able to navigate the small branches there was vital. And that gives us a clue as to why their hands were so different from what came before.
Creatures with really short fingers and claws, they're really great at crawling these big, thick branches, but not so much when you get to the ends of the branches.
That's because this sort of hand can't grasp. Lengthening the fingers, and better still, adding a divergent thumb, means you can curl your fingers around even the thinnest branches, grasp them tightly, and remain stable. We think that's why the hands and feet of early mammals changed. What you end up with are primate hands, well adapted to moving around the fine branches of trees, just like these monkeys here.
Embedded in our bodies is our distant past. The hand I used to write with, to type on the computer, to throw a baseball. That hand has a long evolutionary history, and one important point in that history was here in the trees, on the fine branches, that made a hand with longer fingers and a thumb.
There was a time when our ancient ancestors' hands had digits that all pointed the same way, tipped with claws. But our primate ancestors changed that. We've lost our claws and now have nails and opposable thumbs that allow us to grab things. What I want to know is when our hands started to become as functional as they are today. And one way to see that is to find out when we started making tools. Ancient tools can be found right here in the Great Rift Valley in Ethiopia. Erosion has uncovered these rocks in badlands that are over 2.5 million years old. Paleontologists Bill Kimball and Don Johansen unearthed fossils of early humans here. They've also found plenty of evidence that those ancient hands were making stone tools.
So what do I look for?
Well, you'll look for flakes on the ground, something that is about this big, slightly smaller perhaps. Oh yeah, you got a nice one.
This is very typically...
A nice one? I don't get this at all.
Yeah, I know. It looks like something from your driveway. Right. Some early human took a stone, a hammer stone, and struck it right there. And got this,
you got
and you see you have this, it's called the bulbar percussion. Yeah, okay. You could use it to strip off meat or whatever.
You see all that in the rocks. It's almost like a time machine you have, the way you see this.
It is. It's a time machine that takes us back to a period when the faintest glimmers of what it means to be human are beginning to emerge. The use of the hand, the cognition, the repeated behavior to produce a stylized tool.
So I've seen an ancient stone tool, and now I want to know about the changes that took place in our hand that allowed our ancestors to make them. So I pay a visit to Tracy Kivel, paleontologist from the Max Planck Institute for Evolutionary Anthropology. I'm hoping to find out how our ancestors became as handy as we are today.
Hello?
Hello.
Hi, Harry. Neil Schumann.
Hi,
Good
Tracy.
to see you.
Nice to meet you.
These don't look like fish.
They're definitely not fish. Yeah.
So I know what that is. I mean, we teach human anatomy. That's a human hand.
Yes, that's a human hand. One of the most important things about the human hand, which allows us to use tools so well, is that we have relatively short fingers and a long thumb. And having a really long thumb allows us to better, move the pad of our thumb towards our fingers in what we call precision grips, like we do when we hold a pen, for example.
Or dialing your cell phone.
Or for texting.
In this box are the fossils.
Yeah, so we see fossils from 4 million years ago up to 1.75 million years. People who do what I do, people who study hand bones, we're interested really in two questions, is what are they really doing with their hands in terms of, are they still using their hands for climbing in trees and for locomotion? And when do they start using their hands for tools?
So who's who in the zoo? What are their names and where are they from?
This first one here is Ardipithecus, found in Ethiopia, and it dates to about 4.4 million years ago.
But we're only seeing parts of 2 fingers there, right? So
Yes.
which parts of the fingers are there? They're here?
Yes, exactly. So this actually fits right here. So it gives you a sense.
Wow, it's really long.
They're very long fingers. And then finally, this hand here is the hand of Homo habilis, or the handyman, which comes from Tanzania, and it dates to 1.75 million years ago. And this is a really important hand in human evolution because it was found with stone tools and was considered to be sort of the first tool-making hand.
By piecing together different fossils of the same species, the bone structures of ancient hands become more clear. The long fingers and thumb that make up the hand of Ardipithecus isn't capable of fine precision grips. But over 2 million years, the fingers shrunk, making it easier for the thumb and fingers to work together. The fingers of Homo habilis are far better adapted for precision gripping and are therefore more likely to be creating tools. So the fossil record tells us that our tool-making capabilities are inherited from Homo habilis, which had the hands for making tools about 2 million years ago.
That's another product core? No. Yes, maybe.
That's a maybe. Looks a little rough.
From my earliest days in the anatomy lab, I've always enjoyed tracing the connections between humans and other members of the animal kingdom. And those ties sometimes present themselves in unexpected places.
When I walked into the anatomy lab for the first time, I was sort of scared about what I was going to see, what I was going to feel. But the reality is, after the first few weeks, that fear turns into a sort of a cocky self-confidence. And these things, when you dissect them, it doesn't look very human. It kind of looks like a wax model in a lot of ways because it's been preserved. But then you hit the hand. And for me, as I unwrapped the gauze and revealed the palm, the fingers, the fingernails, something else hit me entirely. And that was a deep sense of connection, a connection to another human body lying on that slab. This was not a wax model that I was dissecting. This formerly was a person who lived a life just like I am living now.
The structure of the hand is remarkable and complex. Its muscles, tendons, and bones allow us to shape the world around us. But it turns out this basic structure is not unique to humans. In fact, it points to our connections with other animals. By the 19th century, scientists were documenting the similarities among the limbs of vertebrates.
Sir Richard Owen was an anatomist back in the 19th century. He was fortunate to be an anatomist in an age of discovery. And so people were coming back to London with new and oddball creatures for him to analyze. And in analyzing all the different creatures that were brought to his laboratory, he found common patterns.
Although the shape and structure of each limb was very different, Owen started to see an underlying unity. It was as if the same set of bones was being squashed or extended to perform different functions.
Here's a dog. Dogs, run and jump. What do you have? One bone, two bones, little bones, and then the digits, the equivalents of the fingers or toes. And, of course, here's a bird. It flies. Its limb has been modified into a wing, And it has one bone, two bones, lots of bones, and then digits. The amazing fact is, in each of these creatures, the architecture is largely the same as ours. Owen didn't know why creatures had that pattern. It was a mystery to him. It was sort of a mystical concept that there's this common architecture behind all animals. It really took a new insight, an insight from Charles Darwin, which basically said the reason why animals have this common pattern is because at some time in the distant past, they all shared a common ancestor that had a version of this pattern too.
As far back as 375 million years, we've found fossil evidence of a creature that has this pattern. It's fish-like, and it's just beginning to make the move onto land. My team found one of these transitional animals called Tiktaalik after 6 years of exploring an island in the Canadian Arctic. Remarkably, its fin had the beginnings of the one bone, two bones, many bones, and digits pattern. It's good evidence that the basic form of this complex and quintessential piece of human anatomy, the hand, emerged from the fins of ancient fish. Studying the DNA of ancient organisms is a window into our distant past. And if you look hard enough, you can find some creatures today that haven't changed much in hundreds of millions of years. This coast is home to a relative that shows us how ancient the genetic recipe is for building our brain.
Yeah, how do you know where to look? Well, you said you want to go deep into this.
Meet Peter Holland, head of zoology at Oxford University in England.
Right, okay. Well, we could start trying around here. X marks the spot? X marks the spot. Let's give it a go. Right. Okay, so what do I do here? Just shake it. You got one. There's an Amphioxus. We got an... Whoa, look at that little guy go. Hey! Just dip it in the water again, you'll see it. It'll flick around. Well, they really flick. I see. There he is. Hey, that's beautiful. Look at that. Which is the front?
It doesn't look like much, but this tiny creature, called Amphioxus, has much to tell us about our own brains.
That is cool. Okay, that ranks as cool. Okay, so let's take a look at a couple of these.
So I brought this from the anatomy lab in Chicago. This is a human brain. And when I look at this, I don't find any obvious similarities to that.
Deep in the genes of this animal and the development of this animal, and deep in the genes of us and the development of us, there are the similarities. So if you just take a look at that, he's just moved.
Oh, he twitched away. Yeah, he's
pretty active.
Holy cow! Oh, that is beautiful. God, they're so clear too. You can see right through them. Yeah, it's amazing. Yeah.
Amphioxus lives in the sand of the ocean floor, filtering algae out of the water. They have a simple nerve cord that runs the length of the body, which ends with a tiny swelling invisible to the naked eye.
If you look at the front end of this animal, you don't see it all expanded into a large skull or brain region. It's just pointed.
We might not have similar brains, but what we do have are similar genes. To make the forebrain, midbrain, and hindbrain, you need a series of control genes to tell the cells within the developing brain where they are. They map out the brain's 3 basic regions. Versions of the same master genes that help form our own brains are also active in Amphioxus, but here they're simply building the primitive nerve cord and the tiny swelling at its front. This means that the genetic roots of our own brains can be seen in creatures that first arose over 500 million years ago.
I think it gives us a glimpse into where our brains came from, into the basic organization of the brain of our ancestors.
I mean, I find that mind-blowing.
He's jumped off, guys. Sorry.
He's going
we
on
have an escapee, sorry.
On land living.
He's gone rogue. Yeah, he's gone for the next transition.
Exactly. He's in the Devonian now.
You may think you have nothing in common with a creature that lives underwater. But at some point in the distant past, all mammals shared a common ancestor, and that ancestor was a fish. It turns out our shared brain structures are one of the best places to see a remnant of our fishy past.
This is a trip to the beginning. This is as basic as it gets. We're going human fish, represented by a cartilaginous fish, a shark on the right. You see the muscles and nerves and organs inside the shark head. But importantly, you see this yellow tissue. This is the front end of the spinal cord. It's the brain. Doesn't look like much, but it's the shark brain. This is the human brain. This is where it all happens for us, where our memories reside, where our fears, our loves, our hates. It's all in here. The human brain, really at the surface level, looks nothing like what's sitting inside the shark head. Yet when you know how to look, you find a very profound similarity. And that similarity lies in the overall architecture of the brain itself. In the human, we have a forebrain. And that forebrain consists of this folded tissue here, where much of our conscious thought happens. And then there's a midbrain, this walnut-sized area in the middle. And then a hindbrain. That's a fundamental division. We see that in development, and we see that based on where the nerves exit the brain. Well, if you follow the similar nerves in a shark, what you see is sharks too have that same structure. A forebrain, a midbrain, and a hindbrain. There's a fundamental architectural similarity among all brains. The brains of people, the brains of dogs, the brains of cats, the brains of monkeys, lizards, frogs, salamanders, trout, bass, even sharks and fish are similar. Each one of them has a forebrain and a midbrain and a hindbrain, despite the fact that those brains are often very different in function and form.
By comparing these two brains, we can see that the basic structure of our brain hasn't changed much since we were fish. I see ghosts of our ancestors in our bodies today. And a fascinating piece of that past is found in our backs.
One of the reasons why falling on our backs hurts so much is because of our inner monkey. Each of us have a vestige of our tail inside of us. We call that the coccyx. And that sits at the base of our spine. When we fall on that, it really can hurt. The reason why it hurts is because it's not covered by much tissue or muscles or whatever. So it's really kind of naked. So, ouch, thank you, inner monkey.
Although it might be nice to have a long tail to help balance out here, our ancestors lost that appendage some time ago. I want to know when that change occurred.
So this is my local zoo, the Lincoln Park Zoo. I come here with Michelle and the kids all the time. But today we're here to do something special. Look at that tail. So you see tails everywhere here. This is like tail central to you.
It is tail central.
I'm here with anthropologist Holly Dunsworth. She's taking me to the one place in the zoo where you definitely won't find a tail. The ape house.
So this feels like home to you here.
Yeah, it does. It does. They are by far the most like us of any creature on the planet. And it's not just because, you know, they're so social and behaviorally complex, and it's also because of their bodies, of their anatomy.
Apes, and that includes us, don't have tails. It's one of the easiest ways to distinguish an ape from a monkey. Although scientists debate why our tails no longer hang behind us, we roughly know when it happened.
We have pretty good evidence for when we lost a tail from the fossil record for ape evolution from proconsul.
So this is straight from Kenya to here?
Mm-hmm. This is a cast of a very famous specimen of proconsul. It's an early fossil ape. It lived about 18 to 20 million years ago. And this is just one of the many partial skeletons that we've got of this creature.
By finding many similar proconsul fossils, scientists are able to complete a picture of these ancient primates. And a tiny bone from the base of the spine reveals a lot about where its spinal cord ends. The shape is a good indication that proconsul is one of the first ape species without a tail.
That is the last sacral vertebra of a proconsul.
Okay.
And I'm going to try to match it up to the one on this chimpanzee. This little bone here is this part of a proconsul.
Oh, I see. So it's
a smaller bodied animal than a chimpanzee, but you've got that anatomy there, that ridge indicating to you that we're looking at the last sacral vertebra. It's tapered so much that it indicates that what follows would have no tail. If it was part of a tailed primate, it wouldn't be tapered.
So taper, no tail.
Right, exactly.
Awesome.
Proconsul answers an important question. Sometime around 20 million years ago, our primate ancestors lost their visible tails and left us with a little souvenir. By adding this fact to our knowledge of that time, we are one step closer to understanding why that transition took place.
You didn't call once. Now we're gonna go here, ready?
So your back's a real pain. Turns out many back problems are the result of our ancestors' transition from walking on 4 legs to walking on 2. The earliest primates lived in trees and moved around on all 4s, much like modern monkeys. But by 3.2 million years ago, hominids were strict bipeds. Fossils like Lucy, found in the Afar region of Ethiopia, are evidence for this ability. Lucy was most likely using 2 legs to navigate in her environment. But standing upright has its costs. Bruce Latimer, a scientist at Case Western Reserve University, studies the causes and consequences of our bad backs.
The human back is indeed the weak link in the human musculoskeletal system. There's an enormous amount of back problems. We took a skeleton like this that was essentially horizontal. We stood it upright. We've had to change essentially every bone to allow us to do that. And we forced it into this new position. We have a problem going from this kind of animal into that kind of animal, and our main problem is balance.
On all 4s, the weight of our body hangs down from our spine. But turn this body upright, and the weight is all out front. We're out of balance.
And in order to balance, we had to create this curve on your back. And then your head would be back here, wouldn't it? So we had to create another curve here in the middle of your chest, and then your head would be sticking out in the front, so we had to create another curve in your neck. So this S-shaped curve in our spine, and we expect that to hold us up. That is an engineering nightmare.
Our curved spine causes all sorts of problems. The vertebrae that make up the bones of your back and the discs between them are put under a lot of pressure, particularly those at the very apex of the biggest curve, the thoracic vertebrae.
If you take those thoracic vertebrae and you push on them from the ends too hard, what happens is this. You end up with what's called a wedge fracture. And the vertebra in the middle of the curve, it's narrower here in the front. This is the front of the body. This is the back of the body. And what has happened is that curve has crunched it, has literally fractured it. Look at the top of it. You can see that's where the fracture occurred. So these human spinal issues are ancient and a consequence of how we walk.
So when you eat, I bet you're not thinking about your inner monitor lizard. But teeth like ours do show up in the fossil record from a time when our ancestors kinda looked like reptiles.
Every time you bite into an apple, right, that is a very complex behavior. Our front of our teeth, the incisors, bite into that apple, and they do so in almost a perfect way. The teeth occlude in a very fixed pattern, a very precise pattern.
And we can see that pattern emerging 260 million years ago. I'm in a desert called the Karoo in South Africa with fossil hunter Roger Smith. He's found ancient reptile skeletons here that can tell us a lot about where our teeth come from. Roger wants to show me a particularly striking specimen.
Ferocious. Look at those teeth.
Look at that. Now this is the Gorgonopsid. These are the carnivores of that time.
You know, this is a fossil that when you see it, you know exactly how it lived, what it did, you know. This wasn't messing around, this creature. I mean,
absolutely.
It's chewing meat.
Yeah.
This Gorgonopsid might look a bit like a dinosaur, but its teeth tell a different story. A reptile's jaw typically houses identical, peg-shaped teeth used mainly for tearing food. Gorgonopsid's teeth are much more closely related to yours, mine, and Roger's.
Compared to the reptiles before it, there was no differentiation of incisors and canines, but this elongation of the canine is particularly a mammal-like feature. Their front teeth were like little needles that came together. Very, very well adapted for flesh tearing.
So here you start to see differentiation along the tooth row. They're all sort of the same shape, but they're different sizes, specialized in different ways.
Gorgonopsids had a virtual Swiss Army knife of teeth. Their giant canines could pierce tough skin. Incisors could strip flesh off a carcass. And, compared to most reptiles, they could chew more efficiently. Never before had such menacingly efficient eating machines ruled the land. In time, back teeth also became more specialized. By 200 million years ago, mammal-like reptiles like the Trithelodont were also capable of chewing their food. This meant more efficient digestion, which in turn meant more energy to chase prey. And eventually, this specialization led to the crushing molars we see in our own mouths today. Teeth come in all shapes and sizes, and to paleontologists, they offer a unique way to study the distant past.
They don't decompose. Skin decomposes. Organs and brains, soft tissues decompose. Teeth, they're the hardest parts of animals. Because of that, they tend to be preserved in the fossil record more than almost any other piece of an animal's body. So when we look at the fossil record of mammals, it's almost exclusively for certain time periods a fossil record of teeth.
And we can tell a lot from teeth.
One of the great anatomists of all time was Baron Georges Cuvier, who lived in Paris in the mid-1800s, who was remarkably important, remarkably smart, and remarkably arrogant. He had a boast, and his boast was, show me the tooth of any animal, and I can reconstruct its entire skeleton. Turns out Cuvier was pretty much right. And here's a tooth. This is a huge tooth, clearly came from a big animal. Flat surface on top, that animal clearly chews plant material. What is a big animal that eats plants? An elephant. Here's another one. Large canines, big scissor-like teeth in the back. This creature clearly, is built to eat meat. This is a lion. So teeth contain so many answers to the basic biology of creatures.
You see, if you found a good set of fossilized teeth, you know something about what that creature ate. That can yield clues to the habitat they lived in, as well as some of their behaviors. The size and shape of teeth also tell you about jaw, skull, and even body size, adding to the amazingly detailed picture you can paint from just one tooth.